Good morning. My talk today is about tools for rapid identification and integration of QTL genes against Blackleg disease. This is a project that is funded by Canola Cluster. There has been many research on identifying QTL from brassica against Blackleg, and all of this research has been conducted under the field using various QTL resources. The circus graph that in this slide I'm sharing with you is from a recent publication by Jackie Batlis group. They have reviewed the QTL studies in brassica against Blackleg, and this graph shows the location of QTLs that have been reported today on brassica genome and BNAPUS. The graph shows location of dormal QTL as green, non-dormal QTL as red, and QTL that has been identified through GWAS studies as orange. So it shows that really how extensive studies that has been conducted in past decades, and there's several QTLs that have been identified. We have also conducted the QTL mapping of BNAPUS genes. We used two different populations. One of them was a cross between Castle as a resistant parents and Topaz as a susceptible. Another population was Sapphire as a resistant QTL parents and Topaz as susceptible. Both of these populations were tested under the field in Australia, and 4 years of study, several different locations. We published the result of this work in 2016, and we were able to identify several QTL that are consistently coming up in multiple locations. So that were, for Castle, these QTLs were on chromosome A1. They were also on A8, and they were on C6 as well. In Sapphire, we had A1, A9, and C6. So as the circles in this shows, the one that I have marked with a circle, there are QTLs that are common between the two populations of Sapphire and Castle on A1 and C6. And these QTLs were also identified in other studies and in other BNAPUS lines with resistance against Blackleg. For example, A1 is identified in the spectrum, has been reported from Grizzly and Dartmoor. A8 and A9 has been identified from Dartmoor, and C6 from Dartmoor and Aviso. So all of these studies, as I mentioned, has been done under the field. There has been report of setting up a QTL assay under the greenhouse and growth chamber. The approach that has been taken is either cotyledon assay, so infecting cotyledon and leaving the infection to progress, or applying the spore suspension to petiole, or to injecting directly into the base of the stem. We use the cotyledon infection method because we thought that it more closely mimics what's happening under the field. So the way that we conducted our experiment is that we use a virulent isolate of Leptosphaeria that did not induce any race-specific resistance on the cotyledon. We infect the plants at the seedling stage of 7 to 10 days, in cotyledon, and then we leave leave the infection to progress, pathogen goes into the petiole stem, and will leave the plants until full maturity. During this period, we make repeated observation, and we note of any abnormality in growth, any withering, or other sign of growth abnormality, yellowing of the leaves, and so on. And at the end of the growing period, at 8 to 10 weeks, we evaluate the stem lesion by cutting the stems and scoring them. So to test our approach, first of all, we use the Castle resistant parent and Topaz susceptible parents to see how they respond. We use isolate 3R11 that is devoid of the majority of the ABR genes, so it does not induce a response at the cotyledon stage. As you see, 3R11 does not trigger any specific resistance on Castle. We see development of lesion and total collapse of cotyledon. And obviously, Topaz that is susceptible and has no origin and no QTL, you see the same phenotype. The plants where these infected plants were left to grow, Topaz, we see that it is totally collapsed. And on the other hand, we see that Castle is growing normally, producing flowers and seeds, and as is expected, so the adult plant resistance here works under the growth chamber condition. We then apply this approach to map the position of the QTL under the growth chamber using 267 DH line from Castle Topaz population. We tested each line individually, and we also run a SNP genotyping on these lines. We mapped the position of the gene, and we identified one QTL on the A8. This QTL is the same as the one that has been identified for this population under the field in Australia. We then questioned that, is it just 3R11 that triggers the resistance? What would happen if you use other isolate? We use isolate 290, French isolate, GR21, a German isolate, and UK01, and isolate from England. So all of these isolates were tested on Castle and on Topaz. Again, cotyledon was totally susceptible. There was no response in the cotyledon stage. But when we look at the stem, as we see here, Castle is totally resistant. There is no lesion against any of these isolates. And Topaz is susceptible with total collapse of the stem and fully developed lesion. We use the isolate 3R11 to screen 150 lines from germplasm that exists in PGRC, Acknowledgement in Saskatoon. The 150 accession that we use shows a variation in response. We scored the lesion development based on a 0 to 10 score, 0 no lesion, and 10 is 100% lesion development and collapse of the stem. So as you see here, the population shows a variation in response with some highly resistant, intermediate, and some susceptible. We use this population to map the location of the gene. Here is a Manhattan plot from mapping data, and shows that the location of the, again, one QTL on A8, the same as the one that we have identified in Topaz, again, showing that, giving more confidence about the approach that we are using. I should say that we have also given this selected some of this resistant accession, QTL resistant accession that was only we identified against 3R11. We have, in collaboration with Rlm4 in Alberta, we have tested against a mixture of isolate, and I don't have the data here to share it with you, but the data that we have is showing that these lines, they were show resistance as well. There was some of the lines that against 3R11 were resistant, and against the mixture of isolate they were susceptible, but overall we see a variation in response, and we see many QTL in this line as well. That is encouraging. To summarize our work, we have applied a GC-based QTL, validated on a topaz castle population, mapped the location of the gene on A8. We have applied it to 150 accession of B neighbors. Again, identified the gene and mapped the location of the gene. And we have also shown that it is effective against multiple isolate. So, to my knowledge, this is the first report that GC-based or greenhouse-based evaluation of QTL has been mapped, and location of the gene has been mapped in a QTL population. To switch gear here, I would like to, the last couple of slides of my talk, to talk about another project that is funded by Canal Cluster, and that is a genetic dissection of Rlm3479 gene cluster, and developing allele-specific marker for these genes. As we know, there has been quite an intense study on GWAS-specific resistant genes because of their practicality and because of their value in breeding. These genes are generally working as a sort of gene for gene. They conform to gene for gene model, but however, the recent cloning of several R gene and many of the AVR gene has shown that this gene for gene interaction is not strictly as the classical gene for gene, and there are cases that, for example, that we have one AVR gene responding to more than one resistant genes. It triggers resistance against several resistant genes, for example, AVR-LM1 against RLM1 and LEPR3. We have recently cloned AVR-LEPR2 in collaboration with Thierry Roussel's group. In parallel work, they identified AVR-LMS, and we realized that we are working both on the same gene, and we have submitted the paper for this. AVR-LMS, AVR-LEPR2, they are the same allele, and they are responding to RLMS and LEPR2. And we have examples of epistatic interaction, AVR-LM7, suppressing the recognition of AVR-LM3, and also suppressing the recognition of AVR-LM5 now. So these are all deviations from the typical gene for gene. Because of the value of the single R gene in breeding for resistance against Blackleg, the search for additional resistant genes has continued. We have now close to 21 genes. The most recent one being Rlm12, Rlm13, Rlm14, and two genes that we have identified recently in our lab, LEPR5 and LEPR6, both of them being broad spectrum resistance. We have tested them against 30 isolate, and we haven't seen any susceptible isolate so far against this, virulent isolate against these two genes. So our integration lines has been really instrumental for research in our lab and research in labs of our collaborators and our colleague. And also it has been really useful and instrumental for the cumula industry in general, using it in cumula Blackleg test sites as a control. We have published 2 papers on integration line, one of them in 2016, describing the creation of this integration line. And one of them is we use the integration lines to find out at the molecular level what factors are in play to that determines the variation in response of our genes and the effect of the host genetic background also on function of our genes. Because the integration lines gives us a really an excellent tool for this. We have origins, very different origins, same background, and we have the same origins in different backgrounds, for example, in topaz versus WAS. And we have published this data that shows that at least some of the molecular events that involved in this variation in response of origins or effect of the background, effect of the host. So back to RLM3, 4, 7, and 9. They are all on A7, tightly linked to each other. We recently published RLM9, and RLM9 we showed that it is unlike the 2 previous string RLM3 and RLM2 that were RLP receptor-like protein. RLM9 turned out to be a well-associated kinase-like gene. It is the second well-associated kinase-like, at the time at least, that when we published this paper, that was reported from plants for resistance against pathogens. The first one being STB6 from wheat, and RLM9, we cloned the genomic construct, we transferred it to topaz, and as you see here, to WAS, and as you see here, WAS transgenic containing RLM9 are becoming resistant against the corresponding ABR genes, ABR LM59. However, it does not respond to the when the ABR LM59 is not present. We have a complete lesion. ABR LM59 is induced during the infection at 3 days, and then it's tapered off at around 9 days. This almost correlates with the upregulation of the R gene as well, RLM9. We also wanted to clone RLM4 and Rlm7. For this, we use topaz RLM4 integration line, and we use topaz Rlm7. We first sequence Rlm7 and Rlm4 topaz, and we assembled the genome. We then conducted an RNA-seq on topaz Rlm7 and Rlm4. We conducted RNA-seq on topaz itself, and also as a control, we have put topaz leopard 2 and topaz leopard 1 here. As you see, we have a high level of expression during the infection in topaz Rlm7 and Rlm4. No expression almost in topaz, and there is no expression in leopard 1 and leopard 2. We use this RNA-seq data not only to confirm the candidate gene, at least in silico confirmation, and also we wanted to help us with the annotation of the gene and finding the gene structure. We cloned the genomic clone under its own promoter, genomic construct. We show that RLM4 and Rlm7, the same as RLM9, they are responsive to pathogen and they are induced during the infection. We transferred those genomic construct into WAS, And we tested against the series of isolate. We use isolate 2367. This isolate doesn't have any AVR genes that correspond to these R genes. So we see collapse trichotomy due to development of lesion. We transferred AVR-RLM47 into 2367, and as you see here, there is no resistant response to WASTAR or Topaz. There is no resistant response to RLM9. It shows that basically our genes there is no cross reaction here. And we see that we have a resistant response to WASTAR RLM4 as we expect. And as a control, Topaz RLM4 also resistant. We see WASTAR transgenic RLM7 is resistant, and control Topaz RLM7 is resistant as well. We generated a transgenic line 2367 with AVRLM7 only, and we show that the race specificity of the genes that we have cloned, it shows only resistance on Topaz RLM7 as a control and WASTAR RLM7 as a transgenic line. It does not respond to RLM4 as we expect or RLM9. So this clearly shows that we have been successful in cloning these genes, and we have submitted this paper recently, and hopefully we'll be able to share it with you in the near future. With this, I would like to thank you, the people in the lab that did the work. Nick Larkin, that he's not in my lab anymore, and he has his own lab as a research scientist at Canada, Saskatoon, working on pulse genomics and genetics. Farham Haddadi, he did the work that I described on genome assembly, RNA-seq, gene adaptation, and basically defining the boundary of the construct that we needed for transformation. Colin, Elena, and Gort, they did the evaluation of transgenic lines, generation of DNA for RNA, genome sequencing, RNA for RNA-seq, and so on. This work is in collaboration with Isabel Parkin and Ralph Lang, and I would like to acknowledge the funding that we have received through Canada Cluster Project. And with this, I would like to thank you for your attention, and we'll be happy to answer any questions.